Hello all. Today we will discuss about glycolysis. Glycolysis is a sequence of reactions that convert glucose into pyruvate with the concomitant production of a relatively small amount of ATP. It is the initial process of most of the carbohydrate catabolism, and this serves three principal functions. The first one is generation of high energy molecules as energy sources as part of aerobic respiration and anaerobic respiration. Second is the production of pyruvate for the citric acid cycle as part of aerobic respiration. And third is production of a variety of 6 and 3 carbon intermediate compounds which may be removed at various steps in the process for other cellular purpose. This can be considered as an overview of what happens to most of the glucose molecules that enters into our system. And let us see the overall reaction of the pathway. During glycolysis, glucose is converted to pyruvate while generating 2 ATPs. 2 molecules of NAD+ is also getting converted to NADH. The pathway overview can be as follows. 10 enzyme catalyzed reactions is considered to occur in 2 stages. Stage 1, which is considered to be the preparatory stage, and stage 2, which is called payoff stage. During stage 1, which consists of reactions 1 to 5, glucose is phosphorylated and cleaved to yield 2 molecules of GAP, that is glyceraldehyde 3-phosphate, and in this stage 2 ATPs are getting used. In stage 2, which consists of reactions number 6 to 10, 2 glyceraldehyde 3-phosphate molecules get converted to pyruvate with concomitant production of 2 molecules of ATP. The reactions of glycolysis during stage 1 can be considered as follows. Hexokinase reaction, which is the first ATP utilization step. In step 2, we have phosphoglucose isomerase, followed by PFK1, which is for utilization of a second ATP molecule, followed by aldolase reaction and also triose phosphate isomerase reaction. In the preparatory stage, the first one is the hexokinase reaction. During hexokinase reaction, the glucose molecule will get converted to glucose 6-phosphate. And this reaction is catalyzed by an enzyme which is called hexokinase, which utilizes Mg2+. And please note, we have the utilization of ATP here. The ATP is getting hydrolyzed to ADP and the phosphate is getting incorporated into glucose. In step 2 reaction, glucose 6-phosphate gets converted to fructose 6-phosphate. This is an isomerization reaction in which phosphoglucoisomerase plays an important role, and conversion of an aldose into a ketose is taking place here. Step 3, which is catalyzed by a phosphofructokinase 1, is the step where the second ATP is getting utilized. During this reaction, fructose 6-phosphate utilizes 1 ATP molecule, and in the presence of PFK, specifically here it is PFK1 and Mg2+, gets converted to fructose 1,6-biphosphate. The fourth step is catalyzed by aldolase. Aldolase enzyme catalyzes the cleavage of fructose 1,6-biphosphate in the reaction 4 of glycolysis, and this leads to the production of two 3-carbon molecules, specifically two trioses. One is glyceraldehyde 3-phosphane and the other one is dihydroxyacetone phosphate. Let us see the reaction. This is the fructose 1,6-biphosphate which was produced in the earlier reaction. We can see that an aldolase enzyme is present which is converting this 6-carbon molecule into two molecules of 3-carbon trioses. One can be considered as a ketose, the other one as an aldose. Step 5 is triose phosphate isomerase catalyzed reaction. Now during this reaction, we can see that an enediol intermediate can be formed. Say for example, in the earlier reaction, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate molecules were formed. Now during this reaction, we find that these can be interchanged. That is, glyceraldehyde 3-phosphate can get converted to dihydroxyacetone phosphate and vice versa. This is carried out with the help of an enediol formation. Stage 2, payoff phase, will be having reactions 6 to 10, and we'll discuss the payoff stage in the next class. Thank you.